All right, guys, welcome back to Tutoring with Mr. Sorrels. And I don't know if you guys can read this word up here, but that's exactly what we're going to be going over today, mutation and genetics. We got to give a shout out to Coach Lamone for setting up this beautiful board that keeps me on track and lets me know exactly what I need to cover with you guys. So, without further ado, let's get into it. So, let's talk about just the different types of mutations. Up here, we got beneficial, silent, and harmful. So let's start off by saying all mutations are not harmful. Remember that all mutations are not harmful. Some are beneficial, some are harmful. Most of them are neutral. 90% of all mutations are neutral. So they're somewhere around silent mutations. That's where our silent mutations come in. There's two different types of mutations also inside of beneficial, silent, and harmful. We have point, which affect one trait, one gene, one allele, and we have chromosomal, which affect multiple traits and multiple alleles along the DNA sequence. So, knowing this, an example of one type of point mutation would be substitution, the one you guys often hear the most about. And let me show you guys exactly what substitution would be. Thanks again, Coach Lamone, for the wonderful instructional supplies. So, all substitution is I have one blue, one green. Like a substitute teacher, we take her out and put it back together. So now, that's all it is. That's just a substitution. There's no adding one more. There's no taking away one more. There's nothing changing about the structure or how many alleles or how many codons we have. We just substituted one, okay? Gotcha. So that's substitution, and that is going to be a point mutation. So now let's talk about frame shifts. There are two different types of frame shift mutations. One is going to be, I don't know if y'all can see it. Let me stand up. This would be called insertion. This is a frame shift. See how we went from 2 to 1. That would be a frame shift because we moved the structure of our frame. Now, if we went from this to that would be called deletion, which is still a frame shift because we went from 3 to 2. So, yeah, guys, just remember substitution is going to be a point mutation, and insertion and deletion are both two different types of frame shift mutations. Now, let's talk about a couple ones you guys might or may not heard of a whole lot. Let's start off with missense. A missense mutation. That is any mutation that changes the amino acids. Any mutation that changes the amino acids is a missense mutation. Silent mutations. We already talked about them, how 90% of mutations are silent. That means they're not good or they're not bad. What happens during a silent mutation to our amino acids? There is no change to the protein being made, to the protein being produced. The third one is called a nonsense. Nonsense. What do we do when somebody starts speaking nonsense? 9 times out of 10, you tell them, be quiet, shut up, stop talking. It's the same principle here. When we have a nonsense mutation, it's any mutation that lines up in codes for the word stop. Stop. It's going to tell you stop. This is a nonsense mutation. We can't go any further. We can't go past this. 9 times out of 10, we have to go back and look at exactly we got it confused or messed up at, all right? So, missense, change in amino acids. Silent, no change at all. Nonsense, stop. All right, just keep that at the front of your foreheads, all right? Slide with me, slide with me. Now, let's get into a couple root words and Punnett squares at that. So, up here, you guys will hear a lot of homozygous alleles. Those are any alleles with the same letters. Any alleles with the same letters. So, for example, if I have uppercase, right? That would be a homozygous allele. Same letters. Hetero, different. That would be big B and a little b, right? Easy. Dominant. Let's stop right there for a second. Our dominant are always going to be our capital letters, correct? Whenever we see a capital letter, it doesn't matter what combination it might have right here. It might have an uppercase B, it might have a lowercase b, but that dominant always wins, all right? That dominant always wins. This is going to be the trait that shows you the capital letter. That is going to bring out the phenotype. So, remember that the dominant allele is always going to represent. It's always going to represent. Recessive, those are our lowercase letters. Those are our hidden traits. So it would be a little something like that. If we did B's, we would have a lowercase b. I know it's ugly, but just for the sake of time, we're going to rock with it. Our lowercase letters, recessives. Remember, you have to have two recessives, so lowercase, for that phenotype to show up. For any time that we see capital B, lowercase b, it's still going to be dominant. Whenever we see lowercase and lowercase, we know that our recessive is coming out, our recessive gene. Geno, genes. Genes. That's all it is. And our genes are going to be each of our letters, okay? So BB, that would be a genotype. That would be our genes, all right? It goes together. Don't make it more complicated than it has to be. And pheno, that means physical traits. Those are the things that we can see. Where genotypes are the letters, phenotypes are the physical traits, usually describing the characteristics of the genotypes. Here are two examples for you guys. So, this would be the genotype, this would be the phenotype. The letter and the trait. Geno, pheno, genes, physical. All right, keep that in mind. All right, guys, so let's do a Punnett square right quick just to refresh you guys' memory. Coach Lamone already got us a nice, neat one set up already. So, we see that he gave us dollar signs today. Instead of letters, we got dollar signs. So I like it. So, I see that we got one uppercase S, we got a lowercase dollar sign, and we got two lowercase dollar signs, right? This is just a quick refresher for you guys. Try to see if I can get y'all closer up. That's better. So, in my box up here, I got a capital S with a money sign, correct? So we know that this box is go to that and that. Simple. We're taking one from dad. So this is going to be dad's genotype. This is going to be mom's geno. And just like kids, we're gonna get one trait from dad, one trait from mom, and fill in our boxes. So next box, we got a lowercase $ from dad and a $ from mom. So we're gonna have two $signs. Correct? Boom. Check it. Make sure it's right. Cool, we good. We rocking. Next, we go fill in our other boxes. Dad is gonna drop down a capital S for us. And mom is gonna slide over a $ for us. Cool. Capital S, lowercase s. Got one from mom, one from dad, one from mom. Cool. Now we gonna slide moms over one time. Slide mom over a $. Get one trait from dad, drop that $ back down. Now, our Punnett square is complete. So let's go ahead and answer a couple questions about our Punnett square. Let's talk about just the dominants right now. How many dominant traits or squares do we see? 1, 2. So, we got 2 4 are gonna be square. All right. And that's gonna be S with a lowercase s. And when they ask you guys for ratios or percentages, this is all it's asking you. They're gonna just ask you to put the genotype, the ratio, and what the phenotype is. All right, simple. So then we go to our recessives. We see that we got $, in two boxes. So we're gonna do our $signs. Boom, boom. Got our geno. It's gonna be a 2/4 ratio. And that is gonna be a round phenotype. Correct? Easy. So, when it asks us what percentage of the kids will be square, what percentage will be round, we look at our ratio, half of each, 50% square, 50 round. So yeah, guys, that's genotypes, phenotypes, Punnett squares, genetics, and mutations review for you guys. So, go be great. Been a pleasure having you on Tutoring with Mr. Sorrels. Check back soon, and let's go be great Bearcats.